Wondering how I should start this talk, and rather than tackling that huge title, I found a fortune the other day and I figured to extend it just as well. So this reads, ears hear and eyes see. What then does mind do? And that's kind of the basis for my project. We know a lot about how our brain processes senses, but we don't necessarily know how do we learn, and that's what I was looking for. A bit of background. There's the brain. The brain has been evolving for billions of years, and the human brain has about 100 billion neurons. And in that, there's trillions of connections. And different parts of the brain do different things, but the area I'm concerned about is the prefrontal cortex, one of the most evolved parts of our brain. The prefrontal cortex, you see in that red box, there's a bunch of arrows going towards it. The prefrontal cortex receives a lot of sensory information, vision, seeing, spatial information, and this area kind of makes sense of all the senses. I won't get too much into this picture, but you see it has a lot of crosstalks with many other areas of the brain. And in that, it is the kind of center for higher level cognitive processing. So, how is information transferred in the brain? Through neurons. Each of those cell bodies is a neuron, and where they connect, in that red box, is a synapse. Now, this is where information transfer occurs. A zoomed-in view. Neurons communicate through transmitting chemicals to one another. And many chemicals are transmitted through these synapses, but the one that I'm concerned about is dopamine. Dopamine is a chemical, there it is. It's a neuromodulator. And what that means is that when dopamine binds to a neuron, it affects the behavior of that neuron from then on. It's thought to be a learning signal in the brain, in that dopamine is released during a lot of learning processes, and it evokes plastic responses in the neurons. What this means is that it can strengthen preferred inputs. For example, if you shoot a free throw and you make it, dopamine's role there says, hey, do that exact muscle movement again. And likewise, it can also weaken non-preferred inputs, and that if you burn your hand on a hot stove, dopamine's role there says, hey, don't ever do that again. So our question lies in the two types of dopamine receptors. There's two families. There's the D1 family and the D2 family. And for the most part, no one cell has both of these receptors, so they're a bit segregated. And therein lies our question, because the D1 receptor, when dopamine binds to it, it evokes an excitatory response in the neuron. And that neuron is there more likely to activate its neighboring neurons. And when dopamine binds to the D2 receptor, it evokes an inhibitory response to that neuron, and that neuron is less likely to excite its neighbors. So there's our question. How do we generate a coherent behavioral response when we know dopamine is released in the brain when we learn, but it has these two antagonistic pathways? And that is what we're trying to figure out. And here's how we did it. So the goal of our project was to record the activity of D1 and D2 neurons during learning. And the way we did that was with a mouse model. Mice are very easy to breed, and they're able to learn pretty substantive tasks. And the science behind it, I'll get into it in a bit, but through optogenetics and electrophysiology recordings, we're able to record individual neurons at the single cell level, analyze their activity, and then we can correlate the neuron's activity with the mouse's behavior and say, hey, this neuron is responsible for this action. And the behavior that these mice undergo is called the discriminatory learning task, where they're presented, it's an auditory task, they're presented with two tones, two conditioned stimuli. There's a conditioned stimulus plus, or I'll refer to it now as CS plus, and the conditioned stimulus minus, CS minus. And what does that mean? Every day, the mouse undergoes a 1-hour session where it's presented with 400 trials of either tone. The CS plus might be like a beep, and the CS minus is any other frequency, like a beep or a beep. And the mouse learns to discriminate between the two during learning tasks. So here's the kind of setup that we use. The mouse is in a little plastic cage, and its head is fixed so that it's not able to move, because by reducing movement, we're able to reduce a lot of noise in the brain. And up there is an optrode, I'll get into that in a second, but it's what allows us to record what the neurons are doing. And in front of the mouse is a spout, and the spout releases a water reward during the CS+. So the two trials that the mouse can undergo is the CS+ and CS- trial, where at the beginning of the trial, there's a 1-second tone stimulation. So, for example, with the CS+, there would be a beep, and then the mouse has 1 second after it to lick the rod, And if it licks it, it gets a water reward, and it really likes that. And during CS minus, there's a beep or beep, and the mouse can lick the rod, and lick and lick and lick, but nothing ever happens. So it learns to discriminate, hey, I like that beep, but I don't like the other tones. And that's the discriminatory learning. So, on to the science part. The method we use is called Optogenetics. It's pretty cool, kind of cutting edge, and I'm sure you'll hear about it a lot more throughout the conference. It's a two-part word, opto, light, and genetics, genetics. And how it works is that these really amazing scientists found that there was a light-sensitive protein in algae. You know, algae in the ocean, they get light, and that's how they live. And specifically, this protein responds to blue light, and it was an ion channel that opens up. And that's kind of how the algae produces energy. So scientists were thinking, hey, you know, if we take this protein and take the gene from it and put it in individual neurons in the brain, and then we shine lasers on them, we can artificially stimulate neurons. And that's kind of where neuroscience is at now. So with the right combination of neurons, you can activate an entire brain circuit to control specific behaviors like movement, or in our case, learning. And this is the molecular level, but what this actually looks like is you have a mouse, you put an optic fiber in its head and stimulate it. And the one part that we have to do is we have to record the neurons as well. So while we have the optical fiber, we have to record them by putting electrodes as well. So the tool we use is called an optrode. It's an optic fiber and electrodes put together. And this is what it looks like. We make this, it takes about a dozen hours. Pretty intense micro work. But the idea of it is that everything below that line goes into the brain. And down there is the optical fiber and electrodes. And neurons are all surrounding the optrode. And neurons have these electrical signals. And what happens is the signal goes out through the connector through multiple phases of amplifying and gets into a computer where we're able to do the data analysis. And I'd like to zoom in on that part. So this is it. That's where we do the recording. The clear translucent part in the middle is the optical fiber, which carries a laser into the brain to stimulate the neurons. And around it are 16 microelectrodes, like little metal hairs, that are used for recording the neurons. And I've been saying light stimulation, lasers, and you all are probably wondering what exactly is going on. So here's a sample CS Plus trial. The mouse is presented with the tone for 1 second. It has a second to lick the rod for water. But why do we use light stimulation? And remember, with optogenetics, you can individually stimulate neurons. So we wait a while, maybe like 5 seconds. At a baseline period during the behavior, neurons don't really fire because there's no tone or reward around. So you normally expect the brain to be pretty quiet in the prefrontal cortex. And if we looked at some sample neurons, you know, there might be a neuron, each dot is a neuron firing. There's another one, and there's another one. You see, none of these neurons are really firing during the laser. But we can identify a D1 neuron or D2 neuron by artificially stimulating the neurons. So when we record through all these different electrodes and we see, hey, there's a neuron right there that is responding to all these laser stimulations when normally neurons shouldn't be firing, we can do behavioral analysis on it. So those are the methods we use. And the experiment schedule looks kind of like this. It's quite a bit of preparation. You have to spend 6 weeks breeding the mice, and then you do a virus injection to put that protein into the brain, which takes about 3 weeks for the protein to be expressed in the cells. And then you spend about 2 weeks handling the mouse, putting it in the setup. You don't just throw a mouse, head fix it, and then it learns to lick things for water. It takes a little while to habituate it. And then on day 15, we implant the optrode in the brain. And then from then on, the mouse undergoes the discrimination task. It's presented with the 2 stimuli, and it learns to like the CS Plus more. And we record the neuron behavior, and we correlate. We observe how the mouse reacts to CS Plus and CS minus. So what do we find is, I'm going to present a graph. It might look a bit confusing, but we'll break it down. This is the response to light stimulation. What happens is that each horizontal line represents a trial. And that trial is at 8 to 10 second period. As you go up, it's each subsequent trial. And there's 400 trials in the hour. And each red line represents the time that the laser was on. And you see, after each red line, very reliably, this sample D1 neuron responds, it's artificially stimulated, and that's how we say, hey, during this baseline period, that's how we know that this is a D1 neuron. So now that we know this is a D1 neuron, we can correlate its activity with behavior. And we can actually see, hey, what does this neuron care about? Now, specifically with the CS plus response, you see at time 0 when the CS plus is presented, the beep, there's a huge spike. And that, the y-axis is just a ratio. So you can see in over 50% of the trials, this neuron is very active compared to the baseline activity of that neuron. And with the CS minus response, you see that there too, there's a peak this neuron cares about. And with the reward response, 1 second before the reward is when the tones are given, that neuron is firing, and throughout the whole entire reward acquisition, it's still pretty active. So we would say this D1 neuron really cares about stimuli and rewards. We're kind of getting somewhere. Now, what about the D2 cell? The one that is, the D2 receptor is inhibitory. And we identified, this is a very reliable cell. It responds to light stimulation. It's definitely a D2 cell. And we look at the response, there's nothing really there. That peak on the right is an artifact of laser stimulation. When the red line jumps up, that's laser stimulating. And you would expect, of course, the neuron is going to be active during laser stimulation. But for the most part, it's not too active during the CS plus. Maybe a bit of a dip, but nothing too significant. And same thing for the CS minus. There's no real peaks like we saw for the D1 neuron. And same thing with reward, not much is going on. So, what we do is we get all of our cells together, and through a really complicated algorithm of just finding out what they do, we sort them, and we say, hey, we saw a distinction. And the difference is that the D1 neurons respond to stimuli and rewards. The D2 neurons, we only found 2 so far, but we haven't found any significant response to either the stimulus or rewards. And this is just kind of preliminary data because there's only 10 cells total. But for future directions, we need more mice, and of course more data. And we would like to do a histological analysis, and that means we want to know where these neurons go in the brain. Where are they going? Are they going to vision areas? Are they going to motor areas? How are they controlling the brain? And what I think is the coolest part about this is behavioral modulation. So we did the laser stimulation during a baseline period when nothing's really going on. But once we really crack this, you can do stimulation during the tone, and you could possibly make the mouse discriminate between the 2 tones better and learn faster. Or conversely, you could make the mouse learn slower. And I think we're on the tip of an iceberg there, because, of course you're not going to have little kids running around in school with lights in their brain, but it's also the tip of a metaphorical or literal iceberg of the dopamine receptors. To give you an idea of the complexity behind this, that up there is dopamine, that is the D2 receptor, and all of that is the responses in a cell. And I think once we're able to crack that, we can do so much with learning. For example, a lot of behavioral learning disorders like ADHD, schizophrenia, are all related to dopamine dysregulation in the prefrontal cortex. So as we learn more about how dopamine goes through this excitation and inhibition with the D1 and D2 neurons, we can really figure out how does the brain process learning. And with that, I'd like to thank everyone who's made this possible, Sir, Rose Hills, Leah and Eric, for their tremendous support, the Lab, Andre, the postdoc who has helped me throughout this whole time, and the other undergrads, Ken, Michael, and Evie, who have been really great support throughout the whole project. Thank you. I'm sure there are many neurotransmitters involved. Right now, we're really interested in dopamine just because of the antagonistic responses. Glutamate has many, tons of other receptors with many different responses, but here we can just isolate it to 2. And once we crack that, we can definitely go on to other neurotransmitters.
Do you have any hypotheses as to the role of D2, which elicits a negative response to dopamine, which makes no sense to me?
Yeah, so it's really interesting because in this, I could talk about this for so long, but in the superficial layers of your cortex are actually most of the cells with D1 receptors. And on the deeper layers are cells with the D2 receptors. So information usually is processed going up and then coming back down. One thing I think could be a possibility is that in the upper superficial layers of the brain, there's a lot of excitation going on. And then, in order to get down into the deeper parts of the brain, you have to go through the deeper layers, which are the D2 layers. Perhaps a signal might have to be sufficiently excited in the upper parts of the brain, go through the lower parts, get past that layer of inhibition with the D2 neurons, and then get to the rest of the brain transmitting a signal. So it's a very hypothesis, but who knows?
Okay, so I have a question. Yes. So you mentioned the control of behavior, but based on what you saw so far with the behavior of D1 and D2 neurons, what would you expect the results would be stimulating during the behavior itself?
Well, so we saw that the D1 neurons have a big response to stimulus and reward. Now, what could happen is if we stimulate the D1 neurons during the tone, when they are normally active, maybe that evokes a response that says, hey, this is a tone that we really like, and we're going to get a reward from it. And during the early phases of learning, if we stimulate during the CS plus tone, perhaps the mouse can learn to discriminate between the two tones a lot faster.